Aloha, students. Today I am pre-recording this lecture because I have to attend a faculty meeting that's taking place during our normal class time. Okay, so it turns out I think this particular lecture is a good one for you to have a recording of anyhow, because throughout the semester we will talk about lots of pretty complicated cellular pathways involving proteins and receptors and different types of cell-to-cell interactions. But one of the most important set of pathways for understanding how our innate immune system works is what we call the complement pathway. And, the way the textbook is set up, the first couple chapters kind of get through our discussion of innate immunity. And so, unfortunately, the complement pathway is one of the trickiest pathways to kind of visualize and understand. And so the fact that it's, in our third lecture is just kind of an unfortunate timing thing. But that's okay, because as you will find out, it's really not that difficult to follow if you sort of talk your way through it. All right, so that's what we will do today. And you have this recording to help you with that. Before we get into today's lecture, I wanted to remind you that pretty much every week you will have inquisitive homework assignments due, right? If this is the schedule that's available in La Lima under the syllabus tool, it's a reminder that I've listed what chapters are due as homework, what chapters you are responsible for the quiz every week. You will see that next week, Wednesday night, you need to have completed the first two inquisitive homework assignments on chapter 1 and chapter 2. Most weeks during the semester, you only have one chapter to focus on. But because we're just starting off and chapter 1 is kind of like a big overview, right? I expect you to have both chapter 1 and 2 done by next Wednesday night. So inquisitive homework assignments, you access them through La Lima as one of the IDAP tools. Right. And they're always going to be due Wednesdays by midnight, essentially. Okay. Reminder that the way the inquisitive assignments work is you have a whole bunch of questions that you can choose from to answer. Your goal is to earn points by answering questions correctly, and you need to reach at least 1,500 points in order for you to have completed the assignment. And you have to get there by answering at least 20 questions. When you are presented the questions, you can read the question and right away sort of decide, do I know this? Do I feel comfortable about this? And you can adjust on the little point slider how much this question is worth. Maybe it can be worth 30 points, right? But if you always had the questions worth 30 points, you're going to have to answer a lot more questions in order to get up to your 1,500 point target. On the other hand, you can slide the slider all the way up to 100 points if you're really confident about it, right? And then you can get through the homework assignment much faster. You would just need to answer, 20 questions, and you'd already have 2,000 points if everything was 100 points, right? You only need to get to 1,500. All right. So how do you decide your confidence level? Well, each question gives you the link to the ebook, right? So you can click on it and actually read that part of the ebook before deciding how many points you want to wager, right? And then once you've decided that, then you can go ahead and answer the question. It's not timed. That's the great thing about it. So you can take all the time you want reading and deciding, right? And the best part is, even after you've read it and you say, well, I still don't get this question. This is kind of strange. I don't want to answer it. You don't have to answer it, right? Because you just move on to the next question. Right? Because there's a big pool of questions. You do not have to answer all of them. You only have to answer enough to get you to your 1,500 target point, right? The reason I emphasize this is because this might be a very different way of doing homeworks than you're used to. Right? So I encourage you, don't wait any longer. Go ahead and at least take a look and try the chapter 1 IDAP, because many of you have already done chapter 1, and I can see some of you have gotten through it in like 40 minutes. All right. Some of you have taken longer, maybe 75 minutes or more. Right. So you don't want to be surprised next week on Wednesday night when you say, okay, finally, I need to do this homework now. And you realize, wow, this takes forever, right? It's meant to be a process of you working on it day by day, answering a few questions so they don't pile up, right? And like I said, because you don't have to do it all in one sitting, right, you just go ahead, do a few a day, skip around, choose the questions that you want to answer. So you feel like you're well on your way to finishing the assignment. And again, that's important because this first assignment due date, you are responsible for 2 chapters. Right? By now, you should be able to answer and finish all the chapter 1 stuff. Right? And after today, you probably can get started on the chapter 2 stuff, even though the rest of the chapter 2 material we won't get to until Monday. All right. But again, because you can choose which questions to respond to, right, you can get started after today's lecture. The other thing are the quizzes. So pretty much every week, you also have a timed Laulima quiz that you need to get done, right? So I make those due by Thursday night. So it's not due the same night as the homework, right? So you have another day to do the quiz. You find those under Laulima, the tests and quizzes tool, and it's just a multiple choice, 10 questions. You have 20 minutes to do it. I strongly recommend you try the practice quiz first, just to see what the quizzes look like, and also work out any sort of technical problems you have with the Laulima. Okay, so I wanted to emphasize that because I don't want you to put off starting the homework too late, all right? And I wanted to remind you that we do have those quizzes also. Okay. So let's quickly review. We had been talking about the fact that physical barriers of our body's tissues are the first kind of line of defense against pathogens that are either surrounding us and on our skin, preventing them from getting in, or potential pathogens that are inside of our GI tract, right? And remember, when I say inside the GI tract, all I'm saying is, from the mouth all the way through the GI tract, the lumen or the space of that passageway, right, is still technically considered outside of the body. We discussed last time, looking at this figure, how these physical barriers are quite different depending upon what tissues you're talking about. Our tough outer skin, the epidermis, represented in the blue outline here, right, that's very tough and impenetrable. The red linings shown in the cartoon here represent the mucosal layers. These are much more delicate, sensitive membranes, right? And they have to be, because they have to accommodate things like gases going back and forth, oxygen and CO2 from the air we breathe in our lungs, those gases being able to exchange with our blood supply. Likewise, the mucosal lining of our GI tract, right, has to be permeable to things like nutrients in the food that we absorb, needing to be able to pass through it. So consider, these are all examples of physical barriers, right? But in addition, there are also chemical components of these barriers. For example, the sweat, right, that is secreted by sweat glands on our skin contain certain types of antimicrobial chemicals in them that we will learn about. The mucus that lines our mucosal layer and keeps it nice and moist, right, because it is a very delicate layer, that mucus itself can be thought of as a chemical barrier. Right? In addition, there are what we call microbial barriers. And we discussed already, we are covered with commensal bacteria, right? The bacteria that we evolved to kind of expect to have on our skin, and even more prominently, inside of our gut, the commensal good bacteria. And we said that the presence of lots of good bacteria actually helps keep the bad bacteria from multiplying, right? So these barriers then can be physical, they can be chemical, as well as microbial. Right? And all of these sort of work together. Okay. So when you have microbes, such as bacteria or other types of particles like viruses that breach or get through these barriers, right? That's when our innate immune response has to kick in. And again, our innate immune response is our first sort of immune response to infection. Here in the cartoon, you can see you have a wound. Maybe this is your skin, right? Your outer skin, the epidermis, and then the softer skin underneath the tissue, right? In pink here, you have, for example, capillaries, tiny little blood vessels that are crisscrossing under your skin. That's what's represented here, right? So this big white space down here, these are the thin walls of one capillary coming through, and the white space would be the lumen inside the capillary where your blood is flowing by this area. So if you get a cut, and let's say bacteria enters the wound, remember, the moment you get a cut and the bacteria gets in, right, that is the start of infection, even though you might not even know that you got cut, right? So these little hot dog shaped bacteria come in. So there's an immediate response. There are macrophages, right? Innate effector cells that are patrolling the tissue. And these macrophages detect bacteria and they ingest or phagocytose them, right? And to destroy them. And as we learned last time, as bacteria are doing this, they're also sending out little protein signals known as cytokines to kind of alert our immune system that, hey, there has been a breach. And this macrophage is essentially saying, I am detecting the presence of pathogenic signals. Right? So the immediate response is one part of our innate immune system. Right? And that's the part that really takes care of like 99+% of all little infections that you have. If you get a paper cut, and you don't even know you have a paper cut, right? Or even if you do, hopefully by the end of the day, you know, that paper cut is already healed. Why? Because all of the bacteria that entered there were already taken care of by just this immediate response, mediated by macrophages, right? And as we will learn, other types of proteins that kind of help out. The second part of our innate response would be, let's say, you know, the wound was a lot bigger than just a paper cut. Let's say, you know, you're riding your bike or something and you kind of wipe out, right? And you get a big scrape. Right? And so a lot of bacteria get in. Maybe the macrophages that are patrolling around, you know, in your arm, for example, maybe they're kind of a bit overwhelmed by all this bacteria. Right? Because even though the macrophages can successfully go around and eat up this bacteria because it's, you know, maybe common everyday bacteria, so the macrophages are trained to recognize, you know, these features, there's just too much bacteria. So as the macrophages are busy eating, as long as they keep eating and eating and eating, they keep sending out cytokine messages, right? These orange triangles. And as a result, the cytokine messages start building up, flowing through your blood, and other immune cells are recruited to the site of infection to help out. So we call this then the induced response, right, of our innate immune system. Macrophages release cytokines and you get inflammation. Right? So inflammation, you know what that means. That means maybe a day later, the skin is puffy, it's warm to the touch, maybe it's very firm, right? And it's very sore. Right? In the past, maybe that's when you would have said, oh, I have an infection, right? But as I've been teaching you, the reality is no, the infection happened, right, the day before when you scraped your arm and the bacteria first got in. The fact that your immediate response could not take care of it on its own, and now you have to experience an induced response, right, is simply a sign that your innate immune reaction is working, and it's just now moved on to this induced phase of it. So inflammation is an important physiological response, and we will learn more about why inflammation is so important in terms of giving us a strong immune defense. Okay. So monocytes from the blood and neutrophils, right, these other types of leukocytes get recruited to the area. The monocytes are the ones that can then squeeze out of the capillaries and become more macrophages. Right? Remember, monocytes are just the sort of pre-macrophage version that's circulating around until they reach a particular spot move into your solid tissue and become macrophages. Right? Neutrophils are a different type of recruited leukocyte. And as we will learn, neutrophils are very powerful phagocytic cells that do an even better job of destroying bacteria. Okay, most of the time, if your immediate response didn't take care of it, which 99% of the time it does, then certainly the immediate response followed up with the induced response. Most of the time, this is enough, right? So 99 point, I don't know what we're up to now, 99.9% of infections that you deal with on a daily basis, right, are taken care of by these two parts of the innate immune system doing such a good job, right? And that's why you don't even think about the fact that every day in reality, right, you are experiencing numerous infections, right? Infections when not only a paper cut, right, or a little scrape, but think about things like when you're brushing your teeth, right? And maybe you accidentally, because you're talking to somebody while you're brushing your teeth, and you accidentally jam your toothbrush against your gums, and you're like, ow, right? And maybe there's a little bit of blood, right? Hey, that is a moment of infection, okay? Or you're wearing shoes that don't fit very well, and at the end of the day, your feet are really sore, you take them off, and maybe your ankle has been rubbed a little raw, right? That is an infection, right? And again, we don't think about those things as being life-threatening. Why? Right? Because our innate response takes care of them, right? So that we don't even think about the fact that we did, in fact, experience infections throughout the day, right? So most of the time, that's enough, and our adaptive response is not needed. That's how we evolved, right? And we evolved to be so good at this. So let's explore the function of some of these innate effector cells and also proteins. Right? So macrophages, as I said, they engulf bacteria via phagocytosis. I think this is a really cool photo here, microscopic photo, right? Where you have the macrophage here and you have these little green bacteria. Right? And the macrophage, you can see, is clearly bringing the bacteria to it. And this illustration on the right lets you kind of visualize what happens, right? So the bacteria, the macrophage might bump into the bacteria, and as a result, parts of the macrophage's membrane can attach to the bacteria, draw it in, right? And then the bacteria is engulfed in a vesicle. That vesicle then fuses to other vesicles that are inside the macrophage that contain different types of phagocytic enzymes, right? So proteins whose job it is to break down other cellular components. For example, break down and digest the cell wall or the different proteins of the bacteria, right? So we call this sort of fusion of these two vesicles the formation of a phagolysosome. Right? That's the name given to the vesicle that results from two vesicles coming together, one that had like the bacteria in it that was brought in, and the other that had all these wonderfully nasty enzymes and things that the macrophage has prepared ahead of time, right? And now when those two vesicles fuse, right, you end up with this sort of little digestive pocket, okay? So think about this though. If the bacteria are just, if the macrophages are just wandering around in your tissue, then they're really going to sort of by chance only bump into a limited number of bacteria. It's much more efficient if these macrophages can really stick to these bacteria so they can grab them to bring them in. Right? So how does the macrophage actually know, what it's reaching for? Right? What is it attaching to? Because after all, the macrophage is going to be bumping into way more of your normal cells in your body compared to bumping into sort of a bacteria, right? So how come the macrophage doesn't just keep trying to eat like everything it bumps into? Right? So how does the macrophage know where to reach? How does the macrophage know what to grab? This challenge is made even more difficult by the fact that I don't think you'd be surprised to know that bacteria are very good at hiding from immune cells. And so over eons of evolution, bacteria, let's face it, bacteria were here before we were, right? Bacteria have evolved all kinds of surface features that make them very tricky to bind. So for example, gram-negative bacteria such as E. coli have these thick sort of polysaccharide fur coats on them. Here's an E. coli. Let's zoom in on the surface of its outer coating here. And what we see is, of course, the bacteria is a cell. So it's going to have a cell membrane. So that's this bottom part here, the phospholipid bilayer. But above or just outside of its cell membrane, it has this outer membrane that consists of these long chains of sugars, right? So these are polysaccharides. Right? Long chains of sugar molecules that have at their end, as part of them, a lipid molecule. So that's why these lipopolysaccharide molecules, or LPS, right, can essentially be part of the outer surface of the bacteria. And so what you end up with is essentially like a thick coating that hides the surface features of the bacteria underneath. You know, it's kind of like if you have shag carpeting in your house somewhere, right? Like my kids in their room, they have this thick shag carpeting because they love to lay on it and read and play, whatever, right? But, you know, a lot of times they build things like Legos, and little Lego pieces get hidden in the shag carpeting and they can't find it, right? Nobody can find it. And I unfortunately find it when I walk through the room with my bare feet, right? And I step on it, right? So why do these LPS, lipopolysaccharide coats, make the bacteria hard for something like the macrophage or other immune cells to detect? Because, right, our immune system and the ability to detect things like pathogen is all about being able to physically interact with different types of molecules, right, that our immune system recognizes as being something foreign or something that's consistent with being something that a pathogen would present. Right? And so if these molecules are hidden under shag carpeting, right, that makes it really hard for something like a macrophage to be able to successfully attach to this. As an example, right, let's think about this here. Here I have, okay, so I say, what is this, right? You say, well, it's a beach ball, right? Well, yeah, it's a beach ball, but this is a macrophage. Okay, and this macrophage then, as it's patrolling around, might encounter this ping pong ball, which is a bacteria. This is actually kind of a nice demo because the relative size difference between the ping pong ball and the macrophage is pretty much, you know, on par with, you know, what we would actually encounter in your body, right? The size of a macrophage versus the bacteria that it's trying to target. But you can see the surface of the ping pong ball, right? Smooth plastic. It's very hard for the macrophage to attach to, right? So the bacteria kind of like is coming by, the macrophage comes by, and it basically bounces off, right? Because the bacteria has that thick coating that makes the surface difficult to grab onto. Right? So in this situation, what could you do here to make it easier for the macrophage to stick to the ping pong ball? Well, you know, to overcome this challenge, we could actually put something on the ping pong ball to make it sticky so that the macrophage has an easier time. Right? So what is this stickiness? Well, there's a very special set of proteins that are made in your body, right, that essentially do this. And so these proteins put special kinds of marks on the pathogen, right? So what is this special protein? It's a special protein that's called Velcro. No, it's not called Velcro. It's called complement proteins. Okay. And once you have the complement protein attached, right, you can have an easier time of the macrophage being able to grab onto the ping pong ball, right? So what does that require? That requires that the macrophage itself, right, has special types of receptors on it, complement receptor proteins that can recognize complement that's been deposited on a bacteria, right? So it can grab onto it. Okay. So complement proteins tag pathogens for destruction. So it's not just one type of protein. It's not just one complement protein. We actually have a whole family of complement proteins. And these are made by your liver. Right? They're made all the time. It's not like you wait until you have an infection to make complement proteins. No, right? Your liver makes tons and tons of complement proteins. And right now, these complement proteins are flowing through your blood, just ready to kind of help out by attaching and coating things like bacteria. So these complement proteins are soluble proteins. They float through your blood plasma. They coat bacteria and viruses with a strongly recognized signal that cells like macrophages can recognize via complement receptors on the macrophage, right? So the fuzzy part of the macrophage, this is the complement receptor that is the right shape to recognize and attach to, right, the complement proteins of the bacteria, right? And so that's how you end up with this process happening. Okay. So the modes of action of complement proteins are, well, as I just showed you, they make phagocytosis, this process, a lot more efficient. Also, these complement proteins can activate and serve as signals that then float away and attract other things like, for example, the macrophage to reach out and grab at a certain area, right, where the complement-coated bacteria is, and also other immune cells can be attracted by the sort of activity of complement proteins to come to the area and help out. In addition, these complement proteins, you know, the Velcro here, can by themselves disrupt the integrity of the cell wall, of the membrane, of the microbe, leading to just sort of the destruction of the bacteria itself without ever involving another immune cell coming along to help out. All right, so that's what we're going to look at today. So as I said, complement is a system of proteins. There's about 30 different complement proteins involved in our immune system, but the most important one is complement C3. Right? In this cartoon over here, C3 is represented by this sort of blue oval. And the basic concepts involved are that, you know, this blue oval, this complement C3 is made by your liver. Your blood is filled with this right now, moving around and around in your body, but it's in an inactive form. Right? In order to activate it, it has to be cleaved or cut. So represented by the scissor here, the C3 is cut, and therefore you're left with two pieces. A larger piece known as C3b, okay, and a smaller piece, C3a. So the C3a is the little piece that then can float off and serve as an attractive signal for other immune cells to know that something's going on here in this area of your tissue, and they can come help out. The C3b is what ends up being deposited on the bacteria, tagging the bacteria. One thing I want to caution you about, this is a perfect opportunity to say, these cartoons in the book, I like these cartoons because I think they do a good job of balancing simplicity, right, for the purpose of being able to have you focus on concepts, right, with complexity, meaning, as you'll see, some of these cartoons do get pretty complicated, right, but I don't think they're overwhelming. But the downside of that is they can be a bit of a trap, right? Hopefully, all of you realize that an actual complement C3 protein is not a blue egg, right? And that you don't really have a pair of scissors that come and cut it, right? Likewise, it's important for you to realize that the complement protein itself is really teeny tiny small compared to the bacteria, right? Because after all, the complement protein is just an individual protein molecule, right? The bacteria, the outer surface here, right? The whole bacteria is made up of, like, you know, all these proteins. So an individual complement C3b attaching to the bacteria, you would not even see it on this cartoon if it was really drawn to scale, right? But the cartoon blows it up so that it emphasizes the concept. But do not be misled and think that in reality, this size is really physiologically what's happening inside the body. Okay, so consider, how does the C3b attach to the surface of the bacteria? What activates the C3 in the first place? What is this pair of scissors, right? And then how does the C3a that floats away attract macrophages? So C3b binds to pathogen surfaces using a thioester bond, right? And you're like, oh no, it's organic chemistry. Organic chemistry rears its ugly head again. Yeah, okay. Well, hopefully you remember that an ester bond, right, is where you have a carbon and you have the double bond to the oxygen, and then the carbon is attached to two other potential R groups, right? So a thioester is an ester bond that involves a sulfur group in there. That's the thio part of it, right? So here we have a more realistic view of what this complement C3 protein would look like. Those of you who have taken Bio 275, hopefully this type of image is more familiar to you. Those of you who have not might be freaking out right now, right? But there's nothing to freak out about. Remember, this complement C3 is one long polypeptide chain of amino acids. That's what a protein is, right? That then folds up to form a tertiary structure, the correct three-dimensional shape that allows it to function and do what it's supposed to do, right? So on this illustration here, the end terminus of the protein might be this end over here. And if you were to kind of play this game of actually following the line, it's like a little maze or something, eventually you'd realize, oh, this is just one long continuous chain that eventually ends up here. And then you have the C terminus of the protein, right? Okay. Within this three-dimensional folded shape, there is a series of amino acids that actually have this thioester bond in terms of how they are connected, right? So this involves cysteine residues that have the sulfur group in them. So normally this thioester bond is just tucked away inside of the protein, but when the C3 is cleaved, when the scissor comes and essentially cuts the C3 molecule, right, part of it floats away as the C3a part that's now liberated. So that's sort of the red and purple part of the molecule here. And what's left, the C3b, has to kind of rearrange itself, right, to get comfortable again, right, because all the amino acid side chains, they're now sort of like needing to reposition themselves. And what happens is that thioester bond becomes exposed. Right. So this thioester bond is highly reactive. Right? It wants to, or it's very easy for it to form covalent bonds with some other molecule that happens to come by. Right? Much of the time, what will happen is that because this is all floating in water, water will just end up interacting with the thioester bond, and you'll have this hydrolysis reaction that basically neutralizes the thioester bond. Right? So you can see H2O has gotten in here. There's now an H attached to the sulfur and then an extra OH over here. That's it. That's the end of the story for this particular molecule of C3b, right? It's become inactive, okay? And it's just going to float away, and it's not going to be any good anymore for attaching to anything else, all right? And you might say, well, that's a waste. Well, yeah, but remember, your body is constantly making tons and tons of these complement protein molecules, right? So the fact that most of them, if they get cleaved and end up just interacting with water, that's okay. Some of them, though, if the cleaving event takes place with them being very close to the surface of something like a pathogen, then there is a better chance that the thioester bond will in fact form a covalent bond with some molecules on the surface of the bacteria. Okay? And so that then forms a very strong bond, very strong covalent bond that cannot be reversed. And now this C3b is now tagged on the surface of the pathogen. Okay. So there are three pathways that we will discuss this semester for the activation of these complement proteins. Right. So hopefully, even though I didn't really say it outright, hopefully you get the impression that these complement proteins are kind of like sort of one of the most important parts of our immediate innate immune response. right? You have a ton of these complement proteins and they're all ready to be activated and attached to things that are foreign pathogens. Right? So the three different pathways that this activation can happen. We call them the alternative pathway, the lectin pathway, and the classical pathway, right? And as they are arranged here on the cartoon, the alternative pathway is actually the first one that acts, right? The lectin pathway is the second and the classical is the third. Right. They're named this way. It sounds kind of weird, right? Because you would say, why is the alternative pathway the first one to act? Unfortunately, this is just a historic sort of byproduct of the fact that the classical pathway was the first pathway that was actually discovered by us and understood, right? That's why it's now called the classical pathway. Later, we realized, oh, wait, there's this other pathway that actually is the one that acts first. So it was called the alternative pathway. But the reality is it's the first to act. Okay. So let's go ahead and quickly get an introduction to these pathways. We will talk about these a lot more in depth as we move on. Okay. The alternative pathway is where the pathogen creates, the surface of the pathogen actually creates sort of an environment right around it that really encourages more of the complement C3s to activate. So preferentially, those activated complement C3s, because they're really close by to the surface of the bacteria, will stick, right, and start tagging the pathogen for destruction. The lectin pathway depends upon certain sugar groups that are present on the surfaces of bacteria. All right, so the surfaces of the bacteria have all kinds of things on them. I already mentioned lipopolysaccharides, and lipopolysaccharide, right? That's an example of a type of sugar structure. But there's another sugar, mannose, that's present on the surfaces of bacteria that are a good target for our immune system to recognize. So the lectin pathway, as we will learn, involves different receptors that recognize these different sugars on the surfaces of bacteria and use that as sort of the starting point to activate complement proteins. And then the third pathway, the classical pathway, is part of our induced innate immune response, right? That happens after inflammation is happening and different cells and additional proteins are coming to help out in the fight against the bacteria. Proteins such as C-reactive proteins, right, which belong to a group of proteins known as pentraxins. They can attach to bacteria and make it much easier for complement protein to activate and bind. Right. So the important thing is all three of these pathways, right, involve ultimately the activation of complement C3. That's why they all feed into, eventually, this is the goal, is to activate the C3. So let's look at this C3 activation pathway. So again, right, I already warned you, this might seem very complicated, but if you just take it step by step, it's really not that bad. All right, it's just the naming of things can be confusing, but there is sort of a weird logic to it. Okay. Let us start off with the fact that here the yellow is the surface of a bacteria. Right? And here is one of these C3 proteins that your liver made. It was floating around in your blood, and now it's sort of like got into your tissues, right? So the complement proteins from your blood can also make their way out of your blood into your tissues, right? So boy, these complement proteins are everywhere in your body, right? And here's a C3 that's kind of floating by. When you get closer to the surface of the pathogen here, right, there's a favorable kind of environment immediately surrounding the bacteria, because bacteria, as part of being alive, they're doing all kinds of their own living functions. So it's not surprising that the immediate area around a bacteria might have different types of pH conditions or other types of things in them. And those things actually make it easier for the complement C3 to activate. Okay? So step one is that the complement C3 does react with water, right, in a step that begins the activation process. Okay. So this C3 interacts with water to form what we call IC3, right? The C3 is still intact, but because of this preliminary interaction with water, right, it is now kind of prepared to activate. And I said the environment close to the surface of the bacteria encourages this IC3 formation. In fact, the spontaneous rate of IC3 formation happens much higher, closer to the surface of the bacteria compared to the C3 that's just floating around somewhere else, like in your blood or in your tissue. Step 2, the IC3, because its shape is a little different now, because of this interaction with water, it is now the right shape to interact with this other complement protein known as factor B. Factor B, the yellow molecule here, right, binds to the IC3, and that actually changes the shape of the factor B. You can see now the shape has changed a little bit, and you have this little groove formed in it. Okay. So factor B is what we call a zymogen. In other words, it is an enzyme that is inactive, floating around, waiting for something to activate it. In this case, the binding to IC3 is what activates the factor B. That little groove in the factor B is the right shape to attract complement protein D, or factor D. Right? This brown sort of teardrop thing that now fits in the activated factor B. And what is factor B? So I said factor B is an enzyme. It catalyzes a reaction, makes a reaction go much faster. And in this case, factor B is a proteolytic enzyme. In other words, once the factor B is fully activated, okay, which is what happens when the factor B binds to it, because factor B binds to it, and you can see it splits the factor B into BA and BB. Right? This BB, right, now is bound to the IC3, and that is now the right shape together to attract another molecule of complement C3 that was floating by, okay, and it attaches there. And what happens is the yellow BB part of this complex, because it's a proteolytic enzyme, it wants to cleave or cut other proteins. What other protein is it going to cut? Well, in this case, it'll be that second C3 that was recruited there. So it activates the second C3, trimming it into C3A and C3B. Right? And hopefully, because this is happening close to the surface of the bacteria, the C3B, its exposed thioester bond, right, now forms a covalent bond with the pathogen. All right, so yay, right? So now you've successfully planted one molecule of C3B on the surface. What happens next? So what happens next is the next cartoon starts with this C3B already on the surface. So in the next cartoon, this is going to be on the left side now, and then we're going to keep going, right? So now here it is. Oh, sorry, right? I forgot that I actually had slides that talked you through the steps 3 and 4, right? So step 3 was the factor D comes and it cleaves the factor B into BA and BB. The BB part stays attached to the original IC3, right? And we call this complex, this is where things get kind of like, you roll your eyes, you know, it's called IC3BB, right? And this IC3BB is now an activated proteolytic complex, right? It's basically a machine that wants to trim other proteins, right? And because that machine is the right shape to preferentially recruit other molecules of C3, right? Then those other molecules of C3 are the preferred target of this proteolytic activity, and you start getting C3Bs, okay, which is right here, right? Step 4. Right? So what do we call? We actually have a fancy name for this proteolytic complex of the IC3BB. We call these C3 convertases. Right? The ACE part, remember, means that it's an enzyme. Okay, so those of you, again, who took Bio 275, you know that, right? And what does this enzyme do? It converts other molecules of C3 into C3B. So these are called C3 convertases, right? Any complex that can activate and convert a C3 molecule would be called a convertase or a C3 convertase. Okay, so now we said, all right, now we're starting off with that first molecule of C3b that got planted on the surface. Right? What happens next? Well, right, this C3b recruits its own factor B, right? And that own factor B recruits another molecule of factor D, And guess what? It's basically a repeat of that first slide, right? Because now you formed again a C3b BB, right? A C3b BB that's now stuck to the surface, right? And it goes ahead and it attracts another molecule of C3, cleaves it or converts it, and sticks that C3b on the surface, right? And you say, wait, how come it takes two different slides in the slideshow to show the same thing over and over again? There is a critical difference between what I'm showing on this slide and the previous one, right? And that is what? On the previous one, you started with C3 that's floating around somewhere, right? And nothing has actually started tagging onto the surface of the bacteria. When that successful event happens and you get the C3b on the surface, right, this slide shows that even though the same process is going to happen now, the fact that you're starting with the C3b on the surface that you successfully planted there makes a huge difference in terms of the efficiency now, right, of any other additional C3s that are being converted for them to be done so right at the surface of the bacteria. So that really increases the likelihood of these new C3bs being planted onto the surface, rather than if this was still happening, just floating around somewhere, right? So that's what's happening here. The C3b BB convertase complex, right, is converting more C3s that are coming in. And because it's doing so already attached to the surface, those C3bs are far more likely, right, to be attached themselves. And once this convertase complex has cleaved the C3, right, and released the C3b and the C3a, it can just keep going, right? So that's why you have this arrow that feeds back and says, well, then it's free to do it again and again and again. And importantly, what? Each one of these C3bs that get planted on the surface, they are also then enabled to do their own formation of a convertase complex. And so very quickly, right, you started from one C3b molecule being stuck there. Very quickly now you have an explosion, right, of C3b molecules as each one of these that gets stuck on there itself goes to work and starts sticking more C3bs. Right? So we call this convertase process specifically the alternative C3 convertase process, right? Because this way of C3 activating and getting stuck to the surface is part of the alternative pathway of complement activation. Right? Okay. So this leads to rapid and explosively rapid covering or coating of the bacteria, right, in a blanket of C3bs. We call this rapid opsonization. So this term opsonization means the rapid coating of the surface of a pathogen, right, to make it easier for phagocytosis to happen, right? So now you're really coating the surfaces of the ping pong ball with the Velcro, right? So the Velcro is opsonizing the ping pong ball, leading to much more successful phagocytosis. So this uncontrolled chain reaction, or it might seem like an uncontrolled chain reaction, right? How is it actually controlled? What do I mean by controlled? Think about this, right? These are just biochemical reactions taking place between the C3 and the surface of some sort of, cell, like a bacteria, right? What's to stop this from happening to the cells, the normal healthy cells of your tissues, right? What a disaster that would be, right? If there was no way to control this process. Basically, all of your complement proteins that are flowing through your blood and your tissues right now, right? They would start opsonizing, your body's normal healthy cells, leading to their destruction. Right? So obviously there must be mechanisms for controlling this. So these are controlled by different, what we call sort of complement control mechanisms, right? So these mechanisms often are other proteins, and these proteins that serve as these complement control mechanisms, right? So here's an example. Some proteins like factor P or properdin, they actually encourage opsonization to happen. You can see in the illustration here, properdin is this pink structure here. Properdin, if it binds to the C3b-BB convertase complex, it helps to stabilize and keep the yellow part and the blue part together. Remember, because these are proteins that are just kind of interacting just because of their shapes. So in reality, right, the yellow factor BB and the blue C3b, right, it's not like they're glued together, right? They're just the right shape to kind of temporarily fit together. If you can stabilize that and discourage them from floating apart, right, you end up encouraging their combined activity as a convertase. So that's what properdin factor P does, right? So this is a way of increasing or enhancing the ability of opsonization of pathogens to happen. On the other hand, you have factors like factor H, this purple ball here that comes, and if it binds to C3b, it causes the C3b to deform in a different way, where factor I, this green teardrop shape, comes in and actually destroys the C3b. Right? So this is one way that, for example, in our bodies, we can sort of keep tabs or put the brakes on improper opsonization of cells by using these types of inhibitory proteins to disrupt the normal opsonization process. So other examples of these sort of convertase controlling proteins are different proteins that are present already on the surfaces of our own normal body cells, right? That would not be found on something like a bacteria, right? So these include things like this decay accelerating factor. So this purple protein that's already on the surface as a normal part of one of your human cells. Uh-oh, look what happened, right? Accidentally, a convertase complex starts forming on your normal cell, right? Yeah, you don't want this thing to start going to work. And so fortunately, the DAF protein comes along, binds it, and it kicks the BB off, right? Thereby neutralizing the ability of this convertase complex to cause any more trouble, you know, and start recruiting more C3s. Likewise, you have other proteins like this membrane cofactor protein, or MCB, that does a similar type of thing. It recognizes that there is a convertase complex on the surface of the cell, right? It binds to it and it changes the shape and boots off the BB, and it changes the shape of the C3b so that factor I can come in and cleave that, neutralizing it. Okay, so that's why this is a wonderful system, right? Because these proteins are on the surfaces of your cells, but bacteria and other pathogens don't have it, right? So that's why when they start getting coated with C3, right, they don't have these mechanisms to prevent it. That's not to say that bacteria don't have other very clever ways of also interfering with opsonization, right? It's just they don't have these two particular examples of proteins that our bodies have. Okay, so I'm going very long here, but I figure it's kind of okay because this is a YouTube recording. So if you're tired of it, you could have stopped and decided to come back at a certain later time, but I might as well just be very thorough, right? So you have this information. This is the last slide though. So the bottom line is complement greatly increases our phagocytic activity, right? And efficiency of macrophages and other phagocytes to be able to attach to and ingest bacteria. So here's kind of a summary slide here. So complement activation leads to the deposition of C3b on the surfaces of a bacteria. Again, remember that the scale here is really wrong, right? This looks like this bacteria has 3 huge C3bs hanging onto it. Hopefully by now you realize no, right? The whole surface of the bacteria is covered with C3b, and the individual C3bs are so tiny that you wouldn't even sort of be able to see them on this cartoon, right? But the point is, this opsonization process that has happened, the coating of the bacteria, right, is like the Velcro being put on the ping pong ball, because the macrophage, the beach ball over here, has complement receptor 1 on it, right? These would be like the fuzzy little stickers on the beach ball that are designed to be the right shape to recognize and interact with the complement proteins, right? And so that makes it easier for the macrophage to attach and bind to the bacteria, resulting in endocytosis and phagocytosis, right? So the bacteria is then brought in, and then these purple vesicles are the lysosomes that are filled with all kinds of enzymes and things like that. When the phagosome joins with the lysosome, you end up with a phagolysosome, right? And then all of the enzymatic contents of the lysosome are now, dumped onto the bacteria, and the bacteria are then digested. So the effectiveness of opsonization through the activity of complements is the reason that most pathogen invasions are eliminated right from the start of infection, right? It is because you have these complement proteins helping out your macrophages. That is why you never die from a paper cut, right? And in fact, your initial being bothered or annoyed that you got a paper cut, right, is over with, by the end of the day, right? It's because this is happening inside of your tissues, right? And that's why I hope you realize that our immediate response of our innate immune system is designed to be so effective, right? And again, though, that idea of it being a relatively low cost for our bodies to maintain. Because what are we talking about here, right? We're talking about the patrolling effector cells like the macrophages, right? These are our sort of moderately trained patrollers that are going around, right? And they just look for certain types of signals, right? Many of those signals they might see on the bacteria itself, and therefore they can go ahead and just attach to the bacteria and ingest it and eat it, right? But that process is made way, way more efficient, right, and faster if you have this opsonization process, the coating of the bacteria by the complement proteins. And again, right, your liver is constantly just making these complement proteins. So it's something that we evolved to do at a very modest cost, right? It's just one of a constantly produced protein by your body, right? But together, it's so effective. Right? Okay, so that'll be the end of today's lecture. And like I said, the nice thing about the video is, of course, you can go back and rewind and take good notes and understand this, right? So a quick reminder, don't forget to get started on your homework assignment, right? At least you should be able to finish chapter 1, inquisitive, in the next couple days, right? So that way you can even start on the chapter 2 inquisitive assignment, because many of the questions are complement protein related, right? And remember, you can start it and just skip over the questions that you don't feel comfortable with yet, the stuff that we'll talk about on Monday. But this way you don't get into a situation where it's Tuesday night or Wednesday night and you're freaking out because you suddenly realize this is going to take you 3 hours or something to get both inquisitive assignments done, right? And in addition, a reminder, think ahead that you do have that first quiz due next week, Thursday night. All right, so anytime you feel ready to take that, you can go ahead and take that. All right. All right. Well, I hope you guys have a great day. If you have any questions, shoot me an email, right? And I'll look forward to seeing you Monday during lecture. All right. Okay. Bye-bye.